Dear all in the audience and dear all in the Zoom and in the YouTube, I would like to give you a greetings for this afternoon. And first of all, I would like to thank the organizers, our students, to give me this opportunity to represent my research, but not just my research, but research of the many, many PhD students, master students, also the postdoc, which are involved in the studying of the molecular technique and using of the molecular technique for the wildlife ecology management and conservation. And recently, as Urška said, by leading the Horizon 2020 project, we also would like to understand how we can connect and how the citizen science can contribute to the genomic and genetic information. So our field of research is quite broad. We were involved in phylogeny and phylogeography, so it means the distribution of the species, then lots of study which is connected with the management and conservation and connected with the population genetics and hybridization. Recently, we start to work also on immunogenetics and epigenetics, as I mentioned before, also on citizen science and human dimension. So the outline of my today talk, because I do not have a time to represent what we do and the kind of our research, but I would like to focus on a few couple of species and their spatial distribution of neutral immunogenetic variation of the European wildcat, European rhodil species, and alpine chamois. And in the end, I also would like to give you a presentation a little bit about how we can use the impact of the genetic variation of the body mass, which is a really important parameters for the management of the roe deer in Slovenia. So just to give a brief introduction regarding the wildlife. Despite the definition of the game species is quite clear, the wildlife definition is sometimes undefined, but most of the studies we are doing, the definition of the wildlife is connected with the species, with the human are recognized that they have some impact or they have some interest of them. So most of them, this could be economic interest, or they can be an impact connected with, for example, disease spreading. So coming to the question, if we are talking about the wildlife, we need to talk about, of course, recently of the scientific-based management of the wildlife. And this scientific-based management is connected with the four important research questions. First research questions we would like to address in the scientific-based management of the wildlife are impact and interaction with the wildlife. So it means the study of the urban wildlife, the vehicle collision, which is increasing the conflict, forest damages, disease transmission. The second research question, which in this area is the prevention and the control. So we are focusing on the issues connected with the disease surveillance, vaccination of the species, exclusion and fencing. You know that today we're connected with the African swine fever. We need to put a fence to make, in some way, isolate the population which are affected. The third question is connected with the human dimension. So the attitude of the humans toward the wildlife, the economic benefits human has for them, the role of reintroduction and intraslocation, and of course, which is also quite important, is how the effectives are educational program, not just at university, but also the education of the citizens and the general public. But of course, the genetics, I think that the most important impact has in the fourth research question, which is connected with the biology and the ecology of the species. And in this area, we can recognize a couple of important issues which the biology and ecology research can answer regarding the scientific-based management of the wildlife. And you can recognize genetic makeup, so the genetic data, but also using the genetic data, we can study behavior trait, diet, even the spatial distribution, even the sociality of the species. So many, many issues that genetics could give us the answer. But what is the genetics? Why the genetics and maybe today the genomics? There is quite a timeline that we start to using the molecular markers, and we start beginning of before the 1990s, we mostly used the molecular markers. We didn't use the genetics markers, and they were mostly allozyme. And then the technology changed, and then we come to the one gene approach. So we use the genetics of mitochondrial gene, nuclear gene, non-coding region, or maybe a microsatellites, but we are studying maybe one or more, a couple of loci. Then in the 2010, around the 2010, we make this huge jump for the genetics to the genomics. And then we start to use genotyping, so the single nucleotide polymorphism, lots of polymorphism in the genome, so not just one loci, but many of them, which will give us the answer regarding the genomic, and many questions connected with the adaptation, for example, of the population. Even though recently we moved from the single, I would say, short read sequencing to the long read sequencing, which it means that recently we can really estimate the whole genome of the species from the telomere to telomere, which means that in the next couple of decades, there are going to be a lot of data, also this kind of genomic data, which we can use in the wildlife management. Which is also important, and it cannot be forgettable, that the price of the genetics really decrease. So it decreases in the way that also people who are working in genetics and genomics, we can now do this kind of study. They are not so expensive anymore. But not just the technology approach, but also the sampling approach change. So we move a step forward for the invasive sampling, like using tissue samples or blood, to the non-invasive samples. Now we can use hair, we can use saliva, we can use feces to detect it, for example, the species distribution or many other genetic questions. Even though recently we can use the environmental sampling, which means that we use the water, we can use soil, even the air, if we want to answer some question if the species was present or not. Even we go further, even we can try to quantify how many species was, for example, in the water body, even though we do not see them through the genetic data. So, and keeping all of this in mind, I will try to summarize a little bit of our research connected with all of this today, and I will start first the research we published recently is the genetic of the European wildcat. European wildcat is divided in Europe in 5 different biogeographic regions, so it's a Felix silvestris, and these 5 biogeographical regions are probably a consequence of some kind of isolation and during the Pleistocene climate change and the influence of the genetic drift, but maybe also as an influence of the recent anthropogenic fragmentation of the species, because we know that the forest habitats which are suitable for the wildcat are really decrease, are degrade, and maybe this is the reason that we have also this kind of distribution and isolation of the European wildcat. But from the work which was published in 2017 by Matucci and the colleagues, you may notice that this region from the Dinaric Park to the Estonian Park, Sandopinic Mountains, there was still unknown regarding the genetic data. So our approach was, of course, to study what is happening with the wildcat in this region. So we, together with the colleagues from Croatia, Serbia, and Macedonia, we did the sampling of the wildcat population, and we also realized that this population in Slovenia and Croatia represent one genetic cluster. So it means that there is a good gene flow through the Dinaric Mountains between the population in Slovenia and Croatia. A little bit different story is regarding the Serbia and Macedonia, but still you may see there should be a one separate cluster, but we need further sampling because we just have a couple of samples from Macedonia to confirm if this European wildcat population in this region are really represented by one cluster, or is it just maybe a bias of sample size? But what was really important in this paper was that the influence of hybridization. Hybridization is something which is really treated wildcat recently. And what was really alarming for us that in more than 50% of the samples from Serbia, there were hybrids. So there's a huge intense of hybridization between the wildcat and the domesticate in that area. In Slovenia and Croatia is a little bit lower, but still it's a concern. And this is something we would like to estimate in this StepChange project using the citizen science, using people who are going into the nature, just to give an idea regarding the distribution of the wildcat, but also regarding the distribution of the feral cat in the area, because we know that they are increasing there are increasing number of the feral cat we can find today in the also in our Slovenian forest. So so this is just a short introduction regarding the work on the wildcat, and I will move now further to the genetics of the European roe deer. Probably you know all of the species. This is one of the really common species in Slovenia, one of the most important game species, not just in Slovenia, but in whole Europe. In Slovenia, the population size of roe deer is around 200,000 individuals. There is a species is managed, which means that there is an annual cull regarding the 30,000 individuals. But what is really important is this management is adaptive management. This is something which is really unique, maybe in Slovenia, that we manage the population of the roe deer based on the several indicators. There are body mass, traffic collision, damages in the agriculture, and so further on. And coming to this question, the ministry approach us if there is an issue that we can use the genetic data to increase and to get a better scientific-based management regarding the European roe deer. So if you are talking about the genetics, you always need to know about the history of the population. And of course, the Slovenian roe deer in the 19th century, there was a stable population in the center part of Slovenia, at that time known region Carniola, which was the part of the Austro-Hungarian Imperial. And this population in the center part was very good managed, and there we also have a data. So it means that the history of the management in population of the roe deer is really old. There was a small colonies in the south part of the Slovenia and the north part, but they were really poorly managed and almost extirpated in that time. Then at the beginning of the 20th century, because of the intense cutting of the forest, there was increasing of the central population because the more habitat increased suitable for the roe deer, and for this the spontaneous colonization to the south, to the Karst region and the Mediterranean occurred, and this spontaneous I would say colonization even last after the Second World War to the north part of Slovenia. So we have one stable population, and then we have this, I would say, confounder effect in the north and the south. So when we start to think about the genetic of the roe deer, we were really surprised that in Slovenia there was no genetic research before our research. The only research which was done on the European roe deer was research from the colleagues, Plis and Camila and their colleagues from Bieloveža, when they saw that in Slovenia there's a representative of two phylogeographic clades of the roe deer. One was the clade, so you can see here in European roe deer is divided into the three phylogenetic clades, west, eastern, and the central. And these two clades, central and the east, are representing in the country with this, I would say, borderline in the central part of Slovenia. Okay, we have also a mixture in the south. So if you want to study genetics and to use the genetic data in the adaptive management, first of all, you need to know something about the spatial structure and the genetic structure of the species. And in this, when we decide to do that, we use in our research just yearling females. So only 1 year old though. Why? Because females show really higher site fidelity comparing to the male. And if you want to study the spatial distribution, it's important to use the female because they have a higher site fidelity, and you can minimize this influence of dispersion of the male. And then we use a different methodology to get insight into the genetic structure of the roe deer. Even though, because we're working with the practitioners, it's important that the genetic costs are really low, and we develop really fast DNA extraction for the mandibles. Mandibles are regularly collected by Slovenian hunter as, I would say, they use mandible as proof that they really harvest this part, so many animals according to the management plan. Otherwise, the hunting family need to pay a penalty if they do not realize the hunting plan. So there's a lot of mandibles all across collected, and we use them for this study. So what the genetics show? Genetics show that there is a division of the roe deer in Slovenia in 3 important clusters, one in the north and another one in the north and another one in the south. And then we have this mixture in the central part of the country, because as I mentioned before, is a consequence that there was a huge stable population in this area in the 19th century. And we also published this last year. What was this even interesting? Always when you're studying a common species, they're really good model species. Roe deer is really good model species to testing the potential barrier in the landscape. So to test the natural barrier and to test the anthropogenic barriers. And we use our data even to test if there are some barrier to the gene flow for the roe deer in Slovenia. And we realized, unfortunately, the highways are obstacles for the gene flow between the population. We found even here one landscape barrier, and this is probably the reason that you have here the Pohorje Mountains and then the Slovengrad, that's huge urban area here, and probably this connection with the hill and then the urban area is not a common habitat for the roe deer. So it's probably another barrier. But we are talking about management and we are talking about the body mass. And if you have in mind that the Slovenia roe deer fence wide diversity of environmental condition, from this sub-Mediterranean dry climate to open Pannonian region, then to the Alps, to the Dinaric, you should expect that there should be some influence of the life history trait of the species if you have such a different environmental variable. And this was even noticed by our colleagues that the females in the sub-Mediterranean area are lighter and produce less offspring compared to the females from the subpannonian region, which they have in average larger body mass and higher reproductive potential. And they ask us, do you think that there should be some genetic background of these differences? What do they even notice? What is really important, I don't know if you know a little bit about the biology of the roe deer, is the issue that the roe deer is an income breeder. This means that it's not a capital breeder and the body mass doesn't change in the time of reproductive season. In the capital breeding is different. The body mass really decrease in the time of reproductive period. But when the females reach the, I would say, sexual maturity, so in the time they start to produce ova, the body mass is really important. And they find that if the body mass was lighter, so lower, the potential of that female was even lower comparing to the body mass. If the female has a higher body mass, they have higher potential. And of course, offspring, a number of offspring is something which is really important for the management, because you can then give a plan. If you know how many offspring you can expect next year, then you can plan how to manage the population. And then we ask ourselves, is there any genetic background for this? And we try to study individual multilocus heterozygosity. So all the individuals which are involved in our study, we estimate the multilocus heterozygosity, and other figures, you have a body mass. And you can see that these red parts are a little bit more present here, like the body mass in this region are higher. So the individual multilocus heterozygosity, obviously in Pannonian region, is a little bit higher. Not so much what we would expect, because probably the reproductive output does not depend only on neutral loci. It doesn't depend only on dilution. Probably there are other aspects which also influence, like environmental variable, to the body mass. But we tested it, we found a correlation, which was not so significant, but there was a correlation between the body mass and the genetic diversity. Even this correlation was higher when we involve genetic membership coefficient. So when we involve when the individual belongs to different cluster, this correlation was even higher. So there should be some influence of the genetics on the individual body mass of the female in the roe deer. Then we haven't stopped there. We move forward because, as I said, studying the natural genetic diversity is not enough. So we move to adaptive genetic diversity. And in this area, one of the genes which are really good studied are the genes of the histocompatibility complex, MSC genes. And we studied DRB gene, their exon 2. These genes are connected with the, I would say, immune defense of organisms to fight parasites, mate choice, sexual selection, and survival fitness. So many of these life history traits are connected with the MSC genes. What we realize, maybe this map is not so good, but this map is a little bit better. You can see that there is a part of Slovenia with higher allelic richness connected to the other part. So assuming now it still has to be discussed, probably it's a consequence of balancing selection, but we do not know, because I think that we should a little bit increase. There is an issue with the MSC in the roe deer connected with the other deer species, but I do not have a time to show all of this data. And we come now to the last part of my talk, is the genetic of the chamois. Chamois is a really interesting species. Interesting species in many areas. Despite that these are mammals, we still do not have a concordance regarding the taxonomy of this species, despite this is a mammal species. So regarding to the behavior and morphological characterization, we have two species of the chamois in Europe. We have a Southern chamois, Rupicapra pyrenaica, which is divided into the 3 subspecies. And then we have a Northern chamois, Rupicapra rupicapra, which is divided into the 7 subspecies. And you can see that the Southern chamois is occupied Pyrenean Peninsula and Apennine. And then the Northern chamois goes from Alps to the Caucasus and Southeast Asia. So there are many studies were done on the taxonomy and phylogeography and phylogeny of these species, because this is also game species. It's very interesting to human. We can say that it's a keystone species in alpine habitat. And they study many, as you may see, there's a lot of publication come up, and they study a different molecular markers, different approaches. They use a mitochondrial region, microsatellites, intron, melanocortin 1 receptor gene, MSC genes, to estimate the history of the chamois and the phylogeography, and of course the subdivision of the subspecies. But assuming also, if you have a little bit of genetic background, that you really need to know that phylogenetic relationship really depends somehow on the marker you are studying. And these are maybe also the consequence that there are still discrepancy into the chamois phylogeny. Why? The first study by Rodriguez showed that the chamois is divided even though not in 2 species, but just in 3 clades. And they named this clade Western, Central, and Eastern. So this was division regarding the mitochondrial DNA. What was really interesting, if you look at these 2 colors, one belongs to Ornata and another belongs to Carthusiana, and belongs to the same phylogenetic clade. But look at here, Ornata belongs to Southern chamois and Carthusiana into the Northern chamois. So there was obviously not accordance with the behavior and morphological data. What do they, authors said? Probably that there was a consequence of this kind of, during the Quaternary period, in this area, there was some secondary contact, introgression between the 2 subspecies, and maybe this is the reason that the mitochondrial DNA doesn't show the clear picture. They move to, and they enlarge the data set with the microsatellites, and then you can see the microsatellites revealed the same story as a behavior and morphological characteristic. So you see Carthusiana belongs to the Northern chamois and Ornata to the Southern chamois. We even though work further using an MSC data and to see if there are some local adaptation connected with the chamois in the region, and maybe that this marker would be better than the neutral markers. But you can see, we also find a mixture of everything. Only what we can state is the Ornata really show the unique genetic background. So it shows the alleles which are not found in other populations, which is probably a consequence of the long-term isolation of the ornata in the Apennine Peninsula. So this is regarding the issue of the chamois, but still we know that the conservation action depends on the species level or subspecies level. And chamois is the only species which have 2 subspecies in the habitat directive. None other species have a subspecies in the habitat directive except chamois. And these are in the habitat directive are Rupicapra tatrica and Balkanica. And living in the region, when we have a contact zone between these 2 subspecies, is really important to divide where is a contact zone between these 2 species and where the Balkanica is represented, because according to the morphological characteristic, sometimes it's really difficult to distinguish them. And then we use the, of course, genetic markers. And as you may see from this figure, we realize that Dinara Mountains, Biokovo and Prain, so these 2 mountains in Croatia and 1 in Bosnia, they have a Balkanica, and the other mountains, they have an Alpan chamois or Rupicapra. So you can see here, even though the 2 different genetic clusters, which is really nice, divide these 2 subspecies. And we are really proud. I many times tell this story to my students. That using our genetic publication, the Croatian Ministry for the Environmental Protection announced Dinara Mountain as a Natura 2000 for the Balkan chamois. So it means that if you have a Balkan chamois in habitat directive, you need also to have a Natura 2000 for the protection and the management of the species. But what was interesting for us, and because we are talking so much about the hybridization is something which is occurs so often in this period of time, which we call it Anthropocene. It was the finding of the hybridization zone in Velebit Mountain. And what was happened here? Before the Second World War, chamois was present here, and then they think that the northern Velebit that was extirpated, and the chamois was introduced. Couple of individuals from Slovenia, another couple of individuals from Bosnia. And I said before, these are Balkanica and these are Rupicapra. So by humans, we make a new hybrid zone in Velebit. And this was something which was not noticeable by people in the field. You cannot notice this by ecology or any other approaches. The genetics can give you this kind of data. But even we go further and we said, okay, now we have a hybrid zone, but what we have before that? What we have before this introduction, and for this story, we use the historical samples. So we use the trophy samples, which was really difficult to collect, from some private collection in Austria, even New Zealand, and we analyze the DNA from that samples, and you may see here that we show so that almost 70% of in these samples, we show the genetic makeup of the Alpine chamois. So it means before the reintroduction in the Velebit, there was an Alpine chamois. Of course, there is not 100% because there is a huge timeline. These samples are more than 100 years old, and there are some mutation rate in the microsatellites during this time of period. Even we can go further using the genetic data, and we can study the distribution on small spatial scale. And we did it in Kupa River. Kupa River is a border between the Slovenia and Croatia. And we know that recently, unfortunately, because of the large influx of refugees and immigrants to the Europe, the government put here the razor fence. This razor fence was recently even changed by the panel fences, which are even more in ethical concern. And we know that there are already published data that there are effects on the direct mortality in large mammals, in carnivores. We do not know what is happening with ungulate, and still we do not have a data. But what we did, we studied, is the Kupa River genetic barrier, is not the genetic, is a barrier, natural barrier for the chamois? And as you may see, no. So in the both sides of the river, you see the same color. So it's one population and there is a gene flow. Probably the situation would be different in a couple of years if the border fence would be there. So this is something I try to represent you for in this short talk. There's many other examples we are working on, and I would like just to announce one of our talks, which our student will have tomorrow. One of our students, we will represent the data by using ancient DNA to study the European and the steppe bison and the distribution of these species in the Holocene and the Pleistocene in the European region, or even though using ancient DNA to distinguish of the hosts of the, for example, the coprolite, which we know that they are fossilized feces, and we can even give an answer to which host these coprolites exist. Another student working on the environmental DNA. I said before, this is something which moving forward, so it's completely non-invasive. We can use the feces, and we are using the feces of predator to study the population density of ungulates. You cannot use genetic monitoring for ungulates like for carnivores. With carnivores, there are a couple of thousand, but for ungulates like a real deer, I told you before, there are 200,000 of them. So genetic monitoring in this way that you're collecting the feces doesn't work. So you need to get a different approach. And one of these approach is that you target the diet of the predator, and you can see how many they predate, if the bull predate more reindeer, roe deer, or opposite, the golden jackal and the lynx. So there is a many issue the genetic can give you an answer regarding, as I mentioned before, management and also the conservation of the species. So this was, I hope that at least some of you understand my talk and that there was not so much genetic data in it. And I would like to thank you for your attention. And again, thanks to organizers to invited me to give a little bit presentation. As I said before, not just, it's a work of the many, many people which are involved. And I hope in the future there will be more and more students involved also in this kind of research. Thank you.